Okay, let's try and unpack this. Just think about it for a second. Maybe 4, even 5 million different kinds of living things on our planet right now.
That's a staggering number, really.
It is. And the core thing, the single unit that connects every single one from, I don't know, the tiniest bacterium up to a huge blue whale, it's the cell.
Absolutely. And what's really remarkable is that with all that diversity, when you drill down to the basics, to the cells themselves, there's this common language, a shared, set of rules.
Mm-hmm.
And that's what we're diving into today, these fundamental ideas of molecular cell biology. We're looking at material covering, well, everything from how cells are built and work, how they move things around,
Right.
to the clever ways scientists actually study them. And we'll touch on cell communication, the molecular side of diseases like cancer, infection.
And even the big picture, right? How cells fit into the story of life and evolution.
Exactly. It's all connected.
So the goal here, for you listening, is to really get a clear handle on these core concepts. We're aiming for those moments, those aha moments where things just click.
Yeah, without getting bogged down in, super technical terms.
Right. So whether you're just curious about how life works or maybe you need a refresher, this deep dive should really help.
Okay, so maybe the best place to start is, what actually is a cell?
Good question. Think of them like the atoms of biology. They're the basic building blocks, structurally and functionally, for all living things.
The fundamental unit.
Exactly. And what's pretty neat is that some organisms, like lots of bacteria, are just one single cell doing everything. All the life processes in one tiny package.
Unicellular, yeah. And what are those essential jobs that every cell has to do?
Well, first off, energy. They've got to manage energy. That's metabolism. Building things up, breaking things down for fuel.
Synthesis and breakdown.
Yep. Then they need to sense what's going on around them, respond to it, adapt.
And reproduce, obviously. Make more cells.
Crucial. And the astonishing part, for me anyway, is that all this incredibly different life uses such similar biochemical tools. And the genetic code, it's basically universal.
It's like everyone's running slightly different apps, but on the same core operating system.
That's a great analogy. Yeah.
And that shared code, it really hints at that single origin, doesn't it? A last universal common ancestor, LUCA. It underlines how interconnected everything is.
Absolutely.
But one of the first hurdles in even knowing about cells was just their size, right? We couldn't always see them.
Exactly. You just can't see them with the naked eye. Our eyes have a resolution limit, maybe 0.1 millimeters. Most cells are way, way smaller.
Like bacteria.
Oh, yeah. A typical bacteria might be 1 micrometer. That's 1,000th of a millimeter. Even our own skin cells are only around 30 micrometers.
And you need help. Microscopes.
Definitely. The light microscope was the first breakthrough for seeing cells at all. But to see the tiny machinery inside the cell, well, then you need the big guns, the electron microscope.
Different tools for different scales. Like telescopes for planets versus distant galaxies.
Like scale comparison.
And the story of how we first discovered them is, well, it's quite a journey. Starts back in the 1600s.
It does. Robert Hooke, 1655. He's the one who coined the word cell. He was looking at cork, thin slices of it under an early microscope.
And so?
Little empty boxes. They reminded him of the small rooms, the cella, where monks lived. So, cell, meaning a little hollow space.
Ah, okay. Then fast forward a bit to the 1830s.
Right, Robert Brown. He spotted something distinct inside plant cells, the nucleus. He called it the nucleus. And the stuff filling the cell, he called that protoplasm.
Protoplasm. And later they distinguished the nucleus contents as karyoplasm.
Exactly. But the big conceptual leap, the formal cell theory, that came a bit later, around 1839. Schleiden and Schwann.
Okay. And their big idea was?
Super fundamental. All living things are made of cells and the products of cells. That was revolutionary.
Changed everything.
Yeah.
And then another piece was added.
Yeah, 1855, Rudolf Virchow. He gave us that famous Latin phrase, omnis cellulae et cellula.
Meaning?
All cells arise from pre-existing cells, which basically put an end to the idea that cells could just, pop into existence spontaneously.
Right. No spontaneous generation for cells. They have to come from other cells.
So you go from seeing empty boxes in cork to realizing all life is cellular and comes from other cells. That's quite the intellectual journey.
Absolutely. And modern cell biology just built right on top of that foundation, especially at the molecular level. Some huge discoveries followed.
Oh, definitely. Like Gregor Mendel, 1865. His work on pea plants laid out the basic laws of heredity, how traits get passed down.
Even though he didn't know about DNA, his insights were profound.
Exactly. And then, just a few years later, 1871, Friedrich Miescher, isolates this stuff from white blood cells, calls it nuclein.
Which turned out to be?
DNA, deoxyribonucleic acid. Imagine figuring out there's an actual molecule carrying the inheritance instructions.
Must have been mind-blowing, and that really paved the way. Thomas Hunt Morgan later showed that chromosomes, those things in the nucleus, were where Mendel's heritable factors actually resided.
Right, linking the abstract idea to a physical structure. But then, the really iconic moment, 1953.
Yeah, yes. Watson and Crick.
James Watson, Francis Crick, building on work by Rosalind Franklin and others, of course. They figured out the structure of DNA.
The double helix.
The double helix.
Yeah.
And it wasn't just the shape, it was how it worked. Their model immediately suggested how DNA could copy itself.
Yeah, that copying mechanism was revolutionary. It finally explained at a molecular level how genetic information gets passed on so faithfully.
It really tied so much together.
And underpinning all of this, there's that quote from Theodosius Dobzhansky.
Oh, the famous one. Nothing in biology makes sense except in the light of evolution.
Exactly. It highlights how crucial evolution by natural selection is. That and the central dogma of molecular biology, DNA makes, RNA makes protein.
Right, the flow of information.
Those two ideas, evolution and the central dogma, they're like the twin pillars holding up modern biology. They make sense of both the diversity and the underlying unity of life.
Okay, so we have cells. We understand their basic importance. We know a bit about how we discovered them. But there's so many different living things. How do scientists even start to organize all that? Classification.
Yeah, classification is key. Over the years, there have been different systems. A really influential one was Whitaker's 5-kingdom system.
Okay, 5 kingdoms. Let's see if I remember. Monera, that's bacteria, and blue-green algae.
Right, though we usually call them cyanobacteria now. Yep, Monera. Then Protista.
Protista. That's a mixed bag, right? Like protozoa, amoebas, some algae.
Exactly. A very diverse group, mostly single-celled eukaryotes. Then kingdom fungi.
Fungi. Okay, mushrooms, yeasts, molds.
Simolds, yep. Then plantae.
Plants, algae, mosses, ferns, flowering plants. Pretty much all the green stuff.
A huge and vital kingdom. And finally...
Animalia. That's us.
Yeah.
And insects, fish, worms, sponges. All animals. Metazoans.
Correct. Now, one really fundamental way to distinguish between these groups, especially at a basic level, is how they get their energy.
Ah, okay. Like producers and consumers?
Essentially, yeah. We talk about autotrophs and heterotrophs.
Autotrophs make their own food. Auto means self, right? Like plants doing photosynthesis.
Precisely. Plants use sunlight, carbon dioxide, and water to make their own sugars, like glucose. They convert light energy into chemical energy. Photosynthetic bacteria and cyanobacteria do this too.
They're the base of the food web, energy-wise.
Absolutely. Then you have the heterotrophs. Hetero means other. They can't make their own food.
So they have to eat, like us.
Exactly. Animals get energy by eating plants or other animals, breaking down those carbohydrates, fats, proteins that ultimately came from autotrophs. Fungi are heterotrophs too, absorbing nutrients, and many bacteria are heterotrophs as well.
Okay, so plants make the energy-rich stuff, and heterotrophs consume it. How do heterotrophs actually get the energy out of that food?
The main way, for most, is aerobic respiration. Using oxygen, that's the aerobic part, to break down those food molecules.
Like burning fuel, but very controlled.
A good way to think about it. It releases the stored chemical energy, and the main waste products are water and carbon dioxide. It powers most animal life, fungal life, and many microbes.
Got it. So that difference in getting energy is key. But there's an even more fundamental split in the types of cells themselves, isn't there? Prokaryotes and eukaryotes?
Yes, that's arguably the most fundamental division in all of biology. And it comes down to the nucleus.
Karyon, the Greek word for nucleus, right?
Exactly. Prokaryote means before the nucleus. Eukaryote means true nucleus.
So prokaryotes don't have one.
Correct. The prokaryotes are just that Monera kingdom bacteria and cyanobacteria. They lack a true membrane-bound nucleus. Their genetic material, usually one circular DNA chromosome.
Just floats around inside.
Well, it's located in a specific region called the nucleoid, but there's no membrane separating it from the rest of the cell's contents, the cytoplasm.
Okay. What else is different?
They divide differently a simpler process called binary fission, basically splitting in two. They also lack other complex membrane-bound organelles like mitochondria.
Where energy production happens in our cells.
Right. In prokaryotes, those functions like respiration or photosynthesis happen mainly at the plasma membrane, the cell's outer boundary membrane, and no nucleolus either.
And eukaryotes, that's everything else. Protists, fungi, plants, animals.
Yes. All eukaryotes do have that well-defined nucleus with a proper nuclear envelope surrounding the DNA, like a control room.
Much more organized internally. And the thinking is that prokaryotes came first.
That's the evolutionary consensus, yes. Prokaryotes are considered the ancestors of eukaryotes, the first forms of cellular life on Earth going way back.
Amazing. And even though they seem simpler, prokaryotes are incredibly successful, aren't they?
Oh, absolutely. They are masters of adaptation. You find them in environments that would kill most eukaryotes instantly. Boiling hot springs, super salty lakes, deep sea vents, acidic mine drainage. They're everywhere.
Tough little guys. But besides being survivors, they actually do a lot for the planet, right? Kind of the unsung heroes.
Definitely unsung heroes. Their collective impact is enormous. Think about primary production, like we mentioned with photosynthesis.
Capturing sunlight energy?
Yep. Turning light into chemical energy, inorganic stuff into organic matter. That forms the base of countless food webs. And crucially for us, oxygen production.
How much oxygen do they make?
Estimates vary, but it's thought that photosynthetic microbes, mostly cyanobacteria in the oceans, produce well over half of the oxygen in Earth's atmosphere.
Wow. Over half. From these tiny things.
Incredible, isn't it? And that's not all. Nutrient cycling is another huge one.
Like recycling elements.
Exactly. Nitrogen fixation is a classic example. Certain bacteria can take nitrogen gas, N2, from the air, which most organisms can't use.
And turn it into ammonia, NH3.
Right, a form that plants can use. It's absolutely vital. They also cycle other elements like sulfur, iron, carbon, and they're master decomposers, breaking down dead organic matter, returning nutrients to the ecosystem.
Breaking down cellulose, things like that.
Yes, breaking down tough plant material, all sorts of complex organic molecules. Essential for keeping nutrients flowing.
Okay, so huge ecological roles.
Yeah.
But they affect us more directly too. Food, for instance.
Oh, yeah. Fermentation, powered by microbes, gives us bread, beer, wine, yogurt, cheese. The list goes on.
So thankful for yeast and bacteria there.
Absolutely. And industrially, they're used to produce vitamins, amino acids, enzymes, all sorts of things.
But there's a flip side too, isn't there? Medicine and disease.
A huge flip side. On the plus side, many of our essential antibiotics were first discovered in microbes. They use them to fight each other off.
Nature's arms race.
Pretty much.
Right.
And we have beneficial bacteria living in us, like in our gut, making vitamins, helping digestion.
Especially for animals that eat grass, like cows. They need bacteria to break down cellulose.
Essential for them.
Yeah,
But
that's
then
fair.
the pathogens. History is full of devastating bacterial diseases.
Like the bubonic plague.
The Black Death, yes. Terrible infections in wartime, childbirth fever before we understood germ theory. And today, things like TB, cholera, STDs, food poisoning, many are caused by bacteria.
So a real double-edged sword.
Okay.
To make this more concrete, maybe we could look at a specific example, like E. coli.
E. coli, Escherichia coli, is a perfect example. It's probably the most studied bacterium on the planet.
Why is it studied so much?
It's easy to grow in the lab, divides really quickly.
How quickly?
Under ideal conditions, lots of nutrients, it can double its population in just 20 minutes. That's its generation time. In standard lab conditions, maybe more like 60 minutes. Still fast.
Okay, fast growth. What does it actually look like?
Structure.
It's got a strong outer cell wall, gives it shape and protection, made of polysaccharides, proteins, lipids, and it has little channels, porins, to let small things through.
And inside the...
Just inside the walls, the periplasmatic space,
Mhm.
then the plasma membrane, that's the main barrier controlling what gets in and out, lipoprotein layer.
And E. coli is gram-negative. What does that mean again? The gram stain. It's a staining technique. Gram-negative bacteria, like E. coli, have a specific cell wall structure that doesn't hold on to the purple gram stain. Gram-positive bacteria do. It's a basic classification method.
Okay. Now, the DNA, it doesn't have a nucleus, so where's its chromosome?
It has one main chromosome, a single circular molecule of DNA. It's surprisingly long. If you stretched it out, it'd be about a millimeter.
Tacked into that tiny cell.
Exactly. It carries maybe 2,000 to 3,000 genes, coding for all the essential proteins, and it's attached to the plasma membrane.
How does that help it divide?
When the DNA copies itself before division, the two copies attach near each other on the membrane. As the cell grows and the membrane sort of pinches inward, it pulls the two DNA circles apart, so each new daughter cell gets one.
Clever mechanism for separation.
Simple but effective binary fission.
And sometimes they have extra DNA too. Plasmids.
Yes. Plasmids are smaller, separate circles of DNA. They aren't essential for basic survival, but they can carry useful extra genes.
Like antibiotic resistance.
That's a really common and important example, yeah. Plasmids are key to how resistance spreads between bacteria. They're also super useful tools for scientists in genetic engineering.
Okay. And inside the cell, the protoplasm. What's in there?
Mostly ribosomes. Loads of them, maybe 25,000 or 30,000 in a single E. coli cell.
Ribosomes make proteins, right?
Exactly. They're the protein synthesis factories, made of RNA and protein themselves. Often you see them clustered on a messenger RNA molecule, making multiple protein copies at once. Those are called polysomes.
And some bacteria can move.
Some have flagella, yeah. Long whip-like tails for swimming around. Size-wise, E. coli is typical, maybe 1 to 10 micrometers, though some cyanobacteria can get much bigger, up to 60 micrometers.
So that's a typical prokaryote. But biology always has outliers, right? What about the really small stuff? Mycoplasmas, viruses?
Good point. Mycoplasmas are the smallest known bacteria, incredibly tiny, just 0.1 to 0.25 micrometers, and they lack a cell wall, which is unusual for bacteria. Some can cause infections.
And viruses.
Yeah.
Even smaller.
Much smaller, usually 30 to 300 nanometers. And viruses. Well, they aren't really true cells. They're more like pseudo-organisms.
Pseudo.
Because they completely depend on a host cell to do anything. They can't reproduce on their own, can't make energy, can't build proteins. They hijack the host's machinery.
So they're parasites at the molecular level. What about bacteriophages?
Those are just viruses that specifically infect bacteria. Phage means to eat.
Okay, so what makes a virus fundamentally different from even the simplest bacterium?
It's that lack of internal machinery. Viruses have genetic material DNA or RNA, and they can mutate, evolve. But they don't have ribosomes, don't have metabolic enzymes, nothing. They're basically just genetic instructions wrapped in a protein coat.
The protein coat is the capsid.
Yes, the capsid. It's made of smaller protein units called capsomeres, often arranged in a very symmetrical shape, like an icosahedron, a 20-sided shape. Very stable.
And the virus makes the host cell build these capsids.
Exactly. The viral genes direct the host cell's ribosomes to make viral proteins, including the capsomeres, which then self-assemble around copies of the viral genetic material. Outside a host, viruses are basically inert. You can even crystallize some of them.
So the key distinction, a living cell has both the genetic blueprint and the machinery to read it and act on it. Membranes, metabolism, ribosomes.
Right, whereas a virus only has the blueprint and relies entirely on borrowing the host's machinery.
That makes sense. Which then leads to the really big question. Where did the first cells come from? How did life even start?
The origin of life. That's a huge question, looking at the evolution of both prokaryotes and then eukaryotes from them.
We have fossils suggesting prokaryotes were around really early.
Yeah, the fossil record points to prokaryotic life maybe 3.5 billion years ago, which is incredible.
But something must have happened before that to get the first cell.
Right. Before biological evolution, there must have been a period of chemical evolution. The formation of life's building blocks, simple organic molecules, amino acids, sugars, nucleotide bases from non-living chemicals.
Spontaneously forming?
Under the right conditions, yes. And then those small molecules somehow had to link up, polymerize into the big macromolecules we see in cells, proteins, nucleic acids.
What are those conditions like on the early Earth?
Very different from today. The atmosphere was probably reducing lots of nitrogen, hydrogen, ammonia, methane, CO2, water vapor, but crucially, very little free oxygen.
And lots of energy.
Tons of energy. Intense UV radiation from the sun because there was no ozone layer yet. Frequent lightning storms, heat from volcanoes.
So this energy and those gases reacted.
That's the idea. The UV light especially could have driven reactions forming intermediate chemicals like formaldehyde, hydrogen cyanide, and these could then react further, perhaps in the early oceans, to form things like amino acids and simple fatty acids, the very first steps towards life.
It's almost hard to comprehend that journey from simple chemicals to a living cell.
It's an amazing story pieced together from chemistry, geology, biology.
Okay, so let's try and wrap this up. We've journeyed from the sheer number of species to the single unifying unit, the cell.
Right. We've seen that fundamental split between prokaryotes and eukaryotes based on the nucleus.
And explored the huge, often hidden roles that prokaryotes play. Despite their size, they're essential.
Absolutely critical, from oxygen production to nutrient cycling.
We looked at a model prokaryote, E. coli, and touched on the even stranger worlds of mycoplasmas and viruses.
Highlighting that key difference between having just genetic material like viruses and having the full cellular machinery.
And finally, we peered back billions of years to the likely conditions of chemical evolution that must have preceded the very first cells.
It really puts things in perspective, doesn't it? Understanding these basics really is the foundation for grasping almost everything else in biology.
Yeah, it helps you appreciate the complexity, but also the underlying unity and the incredibly ancient history of life.
Hopefully it gives you, the listener, a solid framework.
Definitely. So maybe a final thought to leave you with. We started with simple prokaryotes, and now we have this incredible biodiversity. Considering that journey, what other truly fundamental discoveries about life itself might still be out there waiting for us to uncover? What are we still missing?